Bio30s. So for this lesson, let's do a little coin flip to see what we're going to learn about. So let's go with heads. We learn about a synapse, and tails we learn about a synapse. So let's see what happens. And would you look at that? It's heads. We're going to learn about a synapse. So what a synapse is essentially it's a connection between two or more neurons. So generally when we've talked about neurons, we've talked about them being electrochemical signals, the electro being the action potential side of things, the change in charge from negative to positive back to negative, and the chemical side of things would be the synapse. So with the synapse, there's two or more neurons involved, so we're just going to keep it simple and look at two neurons. One neuron will be sending the signal, one neuron will be receiving the signal. The neuron that does the sending is called the presynaptic neuron. The neuron that does the receiving is called the postsynaptic neuron. So when we look at our synapse, this is a synapse right here. We have our presynaptic neuron here, our postsynaptic neuron here, and if we were to define them based on our neuron diagram, a presynaptic neuron would be the end plate, the very, very end. So you go all the way down the axon to the terminal branches, and the very end plate, that would be your presynaptic neuron. So when the action potential reaches the presynaptic neuron, it's going to cause vesicles, which are essentially just little containers. They're going to be moving towards the cell membrane, so the very, very end of that end plate membrane. And inside of the vesicles are neurotransmitters. These are little chemicals that are going to either excite or inhibit the receiving neuron. You may have heard of some of these transmitters before. We'll talk about a few of them, but you may have heard of things like serotonin, dopamine. These are examples of neurotransmitters. So they're chemicals within your nervous system. So then when the chemical, the neurotransmitter, is released across the synaptic cleft, and notice how the two neurons don't actually touch each other. That's why we can't just send an action potential from one neuron to the next. We actually have to allow the chemical to diffuse across the synapse because the nerve cells don't end up touching one another. As the chemical diffuses across the synaptic cleft, it will bind to ion channels, and it will either open a sodium ion channel or a potassium ion channel. So depending on what the neurotransmitter is meant for, excitatory or inhibitory, that will explain what channel opens. Then when the synapse is over, the connection is finished, the neurotransmitter will be broken down by enzymes and then repackaged into the presynaptic neuron. Now the receiving neuron, you've probably already figured this out before, but the receiving neuron is the dendrite. We talked about this in the first lesson. The dendrite receives the action potential, or receives the neurotransmitter in this case, whereas the end plate sends the signal. So I mentioned that there are two types of neurotransmitters. There's excitatory and there's inhibitory. Excitatory neurotransmitters, like the name suggests, will excite the neuron. It's going to keep the signal going. So for example of this would be if you want to say, swing a baseball bat, you want to make sure that signal goes all the way to all the necessary muscles so you can perform that movement, right? That's an excitatory response. Keep the signal going. So when we go back to our diagram here, if it were an excitatory response, you would be opening up sodium channels. Your sodium channels would open, sodium would rush into the cell, this would create a depolarization, and like we learned last lesson, this would create an action potential. And then away we go with our newly depolarized neuron, all the way down into the axon and down that neuron. So that's an excitatory response. Now an inhibitory response would actually be the opposite. An inhibitory response would be, I want to stop this signal or prevent the neuron from being excited. I want to inhibit this neuron from taking this signal any further. What you would do in that case is you would want to hyperpolarize the receiving neuron. So you'd actually open up potassium gates. So potassium gates would open, and potassium would then leave the cell, making it a bit hyperpolarized, even harder to excite. Now you might think to yourself, okay, why would I want to inhibit a signal? Wouldn't I want my nerve signals always going? Well, think about it like this, right? Number one, muscle spasms, right? You don't want your muscles spasming and contracting all the time. You want to inhibit some of those signals. Number two, a very, very common, probably the most well-known inhibitory neurotransmitter is serotonin. So serotonin is found in your brain. The role of serotonin is calming you down, right? It's regulating your mood and regulating your sleep. So if you want to go to bed and you got your mind, you know, racing a mile a minute, and you're thinking of all these things, you want to calm your mind a little bit, that's where the serotonin is going to come in. It's going to turn off some of those signals, help you relax, and help you sleep. So two types of neurotransmitters. Excitatory will open sodium, continue the signal. Inhibitory will open potassium and prevent the signal from going any further. So here's what a neurotransmitter looks like. This is a very common one as well. It's called acetylcholine. So acetylcholine is a neurotransmitter involved in muscle movement. So if you want to move muscles in your muscular system, neurotransmitter that's involved with that would be acetylcholine. So acetylcholine would cause a depolarization in the neurons required to fire up muscle movement. So it's an excitatory neurotransmitter. So acetylcholine would diffuse across the synaptic cleft, bind to sodium channels, open those sodium channels, and then create a depolarization. So it's an excitatory neurotransmitter. Now once you're done with this neurotransmitter, like I said, you want to break that down with an enzyme. The enzyme we use for acetylcholine is called a cholinesterase. So cholinesterase comes along, chops up acetylcholine, and then repurposes it or repackages it. Now it's interesting here that some neurons, associated with acetylcholine, it's also associated with memory. If your acetylcholine is, too low, it could actually contribute to Alzheimer's disease. So one treatment that they're looking into is what if we actually block the enzyme? If we block the enzyme cholinesterase, we would keep the acetylcholine in the synapse longer, allowing that signal to be sent, and then not having as many memory issues. So that's one thing they're looking into there. So once the neurotransmitter is broken down, we repackage it, we put it back into the presynaptic neuron, and we can reuse it again. And that goes for all neurotransmitters. This is just one example. You don't have to know all the different enzymes for all the different neurotransmitters. You don't have to worry about that. But this is just one example where one neurotransmitter, acetylcholine, creates an excitatory effect. We don't want that going forever, because we don't want our muscles spasming and contracting forever. Eventually we want them to stop. So we get our enzyme to come in, chop up that one. This one is actually broken down into acetyl-CoA and choline. You might remember acetyl-CoA from Bio-20. And then the acetyl-CoA, the choline, repackaged into a vesicle and sent back into the presynaptic neuron. So there are literally hundreds of neurotransmitters in the body. These are just a few of the main ones. I'll give you a few examples of some main ones here. So we just talked about acetylcholine. It is mostly excitatory, but at some points it can actually be a little bit of inhibitory, but most cases it's excitatory. You're going to find it pretty much all over your body, your central system, your peripheral system, and at the neuromuscular junction, so the junction between a neuron and a muscle. Its main function is muscle contraction. That's acetylcholine. Norepinephrine, also known as epinephrine or adrenaline. This is excitatory or inhibitory. We find it in central and peripheral nervous system, and I don't like the term there, wakefulness, but you kind of know what adrenaline does. It basically ramps up your body, alerts you, gets your blood flowing, gets your heart rate going, basically helps you prepare for a fight or flight response. So that would be your neurotransmitter. Interestingly enough, your norepinephrine and your epinephrine, which usually go hand in hand, they are one of the chemicals that are found in both the nervous system and the endocrine system. They're both neurotransmitters and hormones, which is pretty cool. Dopamine is usually excitatory. You've probably heard of dopamine before. You do something you enjoy, you have your favorite food, you watch your favorite movie, you hang out with some friends, you probably get a rush of dopamine. We're found in both systems, central and peripheral, and it's involved with emotions as well as voluntary movement. Serotonin we talked about, mainly involved with sleep, only found in the central system, and it is generally inhibitory. And GABA, really helpful with mood regulation as well as motor behavior, also inhibitory and only found in the central system. Now, if your neurotransmitters are malfunctioning in some way, so for example, you have too much or not enough, this can lead to some disorders. So for example, Parkinson's is actually of inadequate amounts of dopamine. Parkinson's is where you get the muscle tremors and the shakes. So for example, Michael J. Fox and Muhammad Ali both have or had Parkinson's, and this is due to an inadequate level of dopamine. If your dopamine is too high though, it can lead to schizophrenia, which is another disorder. So you want to make sure that your neurotransmitter levels are balanced and making sure they're at the correct levels. Alzheimer's we talked a little bit about, but that's because you have low levels of acetylcholine. So low levels of acetylcholine leading to Alzheimer's. Finally, it doesn't always take one neuron to activate another one. Sometimes we might need 2, 3, 4, even more neurons to activate one. So this is called summation, where 2 or more neurons excite or inhibit another neuron. So what happens here is if it's an excitatory response, we have 2 neurons working together. It's like summing in math, right? You find the sum of something, addition, finding the sum of something, we add 2 or more things together. So I might take neuron A, B, and C, and all 3 of them are working together to create a depolarization in neuron D. So they're all summing together, they're all working together to create that depolarization. Or on the flip side, we have neuron A, B, and C creating an inhibitory response, trying to turn off or prevent neuron D from activating. And then sometimes they might even cancel each other out. So sometimes we get a little inhibitory, a little excitatory, both working on the same neuron, and they end up canceling each other out. So that's it for synapses today. Next lesson, we're going to talk about the parts of the brain. So we'll get into all the different parts of the brain, a little bit about the spinal cord, but mostly parts of the brain. We'll talk about all the different lobes and the functions and what each part of the brain does. So thanks again for watching, and we'll chat soon.